Hi everyone, chapter 15, the chromosomal basis of inheritance. Hopefully you're not sick of DNA yet. We still have a few chapters. And it's funny, I kind of end up reflecting on this most years I teach this class because my grandmother was actually a biology major, and this would have been back in the 1950s probably. And they were just learning about DNA then. And so I think, man, when you're taking this class, there's so much more, right? This book is so much thicker than anything my grandmother would have had to know back then. But it's also cool because we have a good understanding of a lot of things that were part of the mystery still back then. Okay, so in this chapter, what I do want to point out is this is not going to be, I would say, anything completely new. When I look at this chapter, I really see it trying to synthesize what we saw in meiosis in chapter 13 with what Mendel found, which is what we studied in chapter 14. And so this idea is, man, Mendel figured out these laws of inheritance, but they didn't really make sense until we realized that the chromosomes, right, are what is carrying the genes, that genetic information, and therefore the basis of inheritance. And so when we explain meiosis in chapter 13, maybe it's not exactly in order of the history of the subject. Okay, so let's jump in here. So again, when Mendel was studying this, he talked about these hereditary factors, but they were purely abstract, right? Today, we can see that those factors, the genes, are actually located on chromosomes. The location of a particular gene can actually be seen if you look, where's my pen, right? So what they're doing in this image, red is a terrible color. If you see these little, it looks like glowing eyes. We've actually, these scientists have actually tagged a specific gene with a fluorescent dye, and now we can see that this is our homologous pair of chromosomes. They're not right next to each other because they're not undergoing division, but I thought that was pretty cool. Okay, so this is a drawing back in the late 1800s, and so people were starting to figure this out as they used kind of better and better microscopes, and then scientists started realizing, hey, what we're seeing, right? The behavior of Mendel's hereditary factors and the behavior of the chromosomes, there's a lot of parallels here, right? And so it's actually in the early 1900s that scientists began to develop what we're calling the chromosome theory of inheritance and kind of putting those pieces together. And so this is what it looks like. And so notice this figure looks very much like one you would have seen in chapter 13 when we're talking about meiosis. But then it also has the P's in there from chapter 14 from Mendel's work. And so this really is kind of the fusion of the things together. And so they're pointing out here a couple of really of the important laws of things that Mendel discovered. And it makes sense when you put these two together, right? So for example, in that parental generation, that true breeding generation, if you had a yellow smooth seed, right, they would always donate the genes for yellow and for smooth, right? And so we saw that in the F1 generation, the dominant trait always showed up if you crossed a true breeding dominant plant with a true breeding recessive plant. And so then this makes sense, right? Mendel was correct. In the F1 generation, and I know this looks crazy, but just stay with me here, right? Basically, again, this is confirming what Mendel found, right? That he said, okay, there's this law of segregation. Basically, the two alleles for each gene end up separated, right? So R's separate, Y's separate. And he also realized, right, that if it was a non-homologous chromosome, right, so we've got R's and we've got Y's, they're not homologous, those also end up assorting independently. They never follow each other into the gametes. Okay, so long story short, what you end up with is a very calculated turnout because what ends up in the gametes always follows this pattern, right? A quarter of them will have both dominant traits, a quarter will have both recessive traits, a quarter is a mixture this way, and a quarter is a mixture that way. So people started putting these two together and saying, oh, this makes sense. This is why Mendel kept getting these numbers, right? It's because the chromosomes, when they're undergoing meiosis, are following this exact pattern, right? And again, when we look at the F2 generation, remember Mendel always found if you crossed two F1s, right, so those two heterozygotes, you always ended up with this 9:3:1 ratio. And really that is explained, again, by those two discoveries he had, this law of segregation and the law of independent assortment. Okay, so again, just a fusion of the last two chapters we've been looking at. Okay, so in this chapter, they definitely get into some of the history, and you were also watching the gene, so hopefully that is going to help you kind of understand what's going on. So I don't want to get lost in the details of this, but I do want to point out a couple of really kind of impressive scientists who figured out some of these details, and then more importantly, right, that we understand these major patterns in inheritance. So again, in the gene, they're talking about Thomas Hunt Morgan, and he's studying fruit flies. And he really came up with some convincing evidence out of these experiments that chromosomes were in fact the location of Mendel's heritable factors. Okay? And so he's using, it's Drosophila melanogaster, which is this common fruit fly. And as he's looking at all these fruit flies in the lab, it's actually a really good choice of organism. Again, kind of like Mendel, it's probably not purely accidental. It's probably that Morgan knew a few of these things, right? They have lots of offsprings. If you've ever had fruit flies in your house, you know they produce lots of offspring. They can be bred, right? A generation only takes like 2 weeks, so they can be bred really rapidly. Again, you don't have to wait like years if you were going to try to use humans. And maybe most importantly, they only have 4 pair of chromosomes, right? Humans have 23 pair of chromosomes. And so this is a fairly straightforward organism. And so really he starts noticing, and this is kind of maybe strange terminology to folks, this reference of this wild type, and you'll actually see this in some of the genetics questions we do in lab. This idea of wild type, it's just normal. It's what he's seeing in these fruit flies all the time. And then whatever the alternative is the mutant, right? And so now we can see that there's multiple alleles, whatever is kind of that normal wild type and whatever that mutant is. And so here's the famous first mutant he ever discovered, which was a fruit fly with white eyes. So there you go. Keep an eye out for them if you have fruit flies in your kitchen. And so as he starts doing some experiments trying to breed or cross this mutant fruit fly, he notices that sure enough, there's in the F2 generation, there's a 3-to-1 ratio, right? And so this was supporting what Mendel saw and kind of confirming that chromosome basis of inheritance. Now, again, this picture is wild, but one other thing that came up here, right? So he had this 3-to-1 ratio that 3 out of the 4 offspring in the F2 generation have red eyes, and 1 out of 4 has white eyes. The other thing so that confirmed Mendel, but the other thing I wanted to point out here, and this was probably a lucky accident that Morgan found, the only fruit flies that got white eyes were males. And so he was also able to come up with this idea of sex-linked traits that are actually carried on the X chromosome, okay? And so we'll look a little bit at what this sex-linked inheritance does. So here we go. So these are actually, this is a microscopic image of the human sex chromosomes. Notice the X chromosome is huge compared to the Y chromosome, right? And of course, if you inherit 2 copies of X, you're female. If you inherit 1 of each, right, that would make you a male. Now, these chromosomes actually do hang out together during meiosis. There's actually like a little segment at the end of the Y chromosome that kind of matches up. So they do act as homologous chromosomes, even though they look totally different. So really, there is a gene on the Y chromosome. It's called the SRY gene. It's the sex determining region on the Y. This gene is responsible for development of the testes in an embryo. And so this is what's going to kick off that basic development in a male. So any gene that's located on either the X or the Y would be considered a sex-linked gene, but typically we'll call genes on the Y chromosome Y-linked genes because there's actually very few genes. There's like 75 or so that we've discovered on the Y chromosome, because if you think about it, right, you have to, you couldn't put anything on the Y chromosome that was necessary for life. Otherwise, females, right, would have some big problems there. And so the genes that are on this Y chromosome are really specific to the development of males. And so we have X-linked genes on the X chromosome, but typically if you hear someone talking about a sex-linked gene, they mean it's on the X chromosome. Okay, so I did want to point this out too, realizing that, so as I'm talking about patterns of inheritance, we'll generally talk about humans, but I did want to point out that this is not always the way that sexual determination takes place, right? So they made this a little confusing. Remember, you have 46 chromosomes. So when they're saying 44, they're saying, well, you have 44, 45, 46, right? So they're trying to show how you would either become male or female there. Right, so those are the offspring. So again, females are XX, males are XY, but this doesn't happen in every system. And so here they're trying to show you what's referred to as the XO system. So if you get 2 copies of X here, you become female. If you only have 1, you're male, right? It's just for example. Notice, at first when you look at this, you're like, oh, well, they just switched the letters, but it's the same thing as humans, but it's actually not, right? This is different because now the female has 2 different genes, and the male is the one who inherits 2 of the same thing. Okay, so a slightly different way of doing it. And then this one, I'm a beekeeper, so this one always fascinates me. This is where the diploid version of honeybees, for example, so a bee that has 2 copies of each chromosome will be female. So that's all the workers and the queen. If you only inherit 16 chromosomes, if you're haploid, you're male. And so basically the way that happens is if the female lays a fertilized egg, right, that brings 2 sets of 16 together, there's your 32. But if she lays an unfertilized egg, she produces a clone of herself, right? This is asexual reproduction, and that clone becomes a male. Wild, right? Okay. I could talk about that for a long time. Moving on. Okay, so this is kind of what it looks like if you actually set this up in a Punnett square. And we are going to do some of these problems, so we'll have a little bit of practice here. But let's take a look at what this would look like, right? So what they're trying to show you here, make sure I do this right, is, so here we have looks like a male that has maybe some recessive trait, okay? And here we have a female with 2 copies of the dominant trait. So they're showing XX to show that it's female, and then they're putting, whatever alleles she happens to have, where this female down here has 1 copy of the dominant and 1 copy of the recessive. So anyway, what you're gonna do is you split these up in meiosis, right? And you can look at all the possible combinations of what would end up in the gametes, right? And do notice, right, it's obviously only the male that's capable of giving a Y chromosome to the offspring. And so it's always the father that is determining the sex of the offspring, right? So notice what happens here is if, say this is like a mutation, say this is actually something that would make, say dad colorblind. Notice he only has one copy of it. It's not a dominant trait, right? We've written it with a lowercase n, but since a male only has one copy of X, there's nothing to kind of cover over it. And so really anything he inherits on the X is going to show up, dominant or recessive. It doesn't matter at this point. Okay, so what we're going to see here is that if he donates that X chromosome, he is always going to hand off, say, this mutation, right? The gene for colorblindness. But notice anytime he gives the X chromosome to the offspring, they're female, right? And so when he gives the Y chromosome, there is no trace of something like that mutant gene for colorblindness. And so this ends up presenting some really interesting patterns of inheritance. So say this daughter, let's say this is colorblindness, right? So this dad is colorblind. I'll just do CB, right? All of his sons have normal vision. All of his daughters have normal vision, right? But they are carriers of this colorblindness gene. So let's see what happens when she produces offspring. And so here we're going to mate with a normal vision male, right? He's got the dominant trait, the normal trait for color vision. She is going to have half of her eggs with the dominant trait and half of the eggs with the recessive trait, which is for color blindness. So notice what happens. You basically run into a situation where 25% of the females, totally normal vision, right, and both copies of the normal gene. You end up with half the females, again, being carriers of this color blindness gene. But then in the males, right, this is where the color blindness shows up. Only in the males whose mother was a carrier for the trait. Okay, and of course, if that same offspring had mated with a male who was colorblind, right, now, sorry, that was weird. Now we can still have sons that are colorblind, but they've inherited that from mom. And we could have a daughter that's colorblind if she inherits that recessive trait from both parents. So these are a little more complicated than that straight up Mendelian genetics, right? But if you just lay out your Punnett squares, we should be able to get through these okay. All right, but basically what it comes down to, right, for this recessive X-linked trait to be expressed, the female either needs 2 copies, right? She's going to be homozygous recessive, but again, the male only needs 1 copy. And that's why we actually see these X-linked recessive disorders much more common in males, which often confuses people because you're like, it's on the X chromosome, that should be female. But again, males, since they only have 1 copy, these things show up. Okay, so again, I use the example of color blindness, but there's a form, Duchenne muscular dystrophy, hemophilia, some forms of hemophilia show up this way as well. Okay, we've got more light. So let's look at some other examples. This is, to me, still one of the craziest things in genetics. And so this is what's called X inactivation. So you got to think about it this way, right? If a male can be totally fine with only 1 copy of the X chromosome, then a female really only needs 1 copy of the X chromosome. And so in fact, what happens in females, at least in mammals, is that one of the 2 X chromosomes in each and every cell randomly gets inactivated. This takes place during embryonic development. And the inactive X chromosome, so see how it kind of like coils up here? Excuse me, this is what's referred to as a bar body. Did they write that out for you? No. So it's BARR, bar body. And so if a female is heterozygous for a gene that's located on the X chromosome, and she inactivates half of those genes for mom, right, one trait shows up. And if she inactivates half those genes from dad, a different trait shows up. And so that's actually what's going on in this tortoise shell cat. Okay, so some of that pigmentation is from mom and some is from dad. And this actually ends up, you can get some of this, and actually pigmentation is one of the things you can see in humans. There's also been some study whether this is related to some autoimmune diseases, right? If the body doesn't inactivate these X chromosomes more or less evenly, there's this possibility that your immune system suddenly gets surprised when it sees some of the proteins made by mom's, X chromosome, if there's very few of those in the body, or, from dad's X chromosome, if there's very few. Anyway, so this is an important thing that you'll see again. Okay, so again, without getting too crazy, Thomas Hunt Morgan figured out a lot of stuff. So one of the other things that his lab worked on was this idea of what genes were on which chromosome. And I actually have this entire binder, I'll try to remember to bring it to lab, that goes through like each and every human chromosome and is talking about all the different things, particularly these are for diseases, but different things that are on different genes. And so one of the things that Morgan found is that genes that are located on the same chromosome tend to be inherited together. So he called them linked genes. This picture is crazy. Let's look at this one. And this sort of makes sense, right? So if this gene B and gene VG are on the same chromosome, right, it would make sense that they would travel together through meiosis, right? But one of the things, and Mendel noticed this too, right, is that sometimes the offspring, right, this offspring would look just like this parent, right, and this offspring would look just like this parent. But around half the time, right, the phenotype in the offspring doesn't look like either parents. And so they called this idea genetic recombination. And so one of the things associated with this, right, is just that, like, okay, this gene came from dad and this gene came from mom, right, and they may say they were both recessive, right. That wasn't going to show up before in this particular combination. So smooth green didn't show up, right? We could have gotten green and we could have gotten smooth, but to show up as smooth green means there was a recombination. So that's part of it. But then the other part of it, and this is crazy to me, that Morgan figured this out, is that even though some genes are linked, right? He knew they were on the same chromosomes. Sometimes he would end up with offspring that were in combinations that didn't make sense according to Mendel's laws. And so he proposed some process must occasionally break the actual physical connection between genes on the same chromosome. And this is now what we know as crossing over. So if you zoom in here, right, he basically figured out, like, well, wait a second, right? Sometimes, right, we might keep a portion of the chromosome, say with a dominant trait, and a portion with a recessive trait, and they shouldn't have been together. But sure enough, it's called crossing over. And we talked about that in meiosis, like, sure, of course, you should totally know about this. Let's skip that. So again, so he basically figured out the actual way that happened. All right, this is what it all comes down to. So if you find yourself getting lost in kind of the nitty gritty, take a deep breath, find some Khan Academy videos, go over this again, right? But what this really comes down to is this idea that we get this huge variation. So all of this kind of shuffling around of genes that's taking place in meiosis gives us these new combinations of alleles, right? And this provides the huge variety that we see in nature, right? And this then acts as the raw material for natural selection to say, oh, like this moth is camouflaged, right? Or, and those traits are going to pass on. Here we've got potatoes, some of which look like pine cones. I'm like, what is that potato? Anyway, And if you think about it, this variation is huge, right? So even if you just think about independent assortment, right? You have 23 chromosomes and they all move independently of each other into, say, those four gametes that you're producing, right? You could have chromosome number 1 from your mom move into a cell, and then chromosome, how do I say this? Sorry, would it be your mom? Yeah. Okay. So, right, you have all these homologous chromosomes, in fact, 23 pairs of them. So, as you think about them splitting, right, this one goes this way, this one goes this way. Well, what happens when you get to the next set? They might go the opposite way, right? And so you actually end up with the way to calculate the potential number of combinations. You take 2 to the 23rd, right? Because there's two genes, right? So they could go either way, and there's 23 of them. So there's 2 to the 23rd possible combinations, which ends up being some huge number in the, I think, millions. Okay, so there's already a huge amount of variation. And then, right, we said, well, so there's random fertilization. So your 2 to the 23rd meets up with someone else's 2 to the 23rd. And now we're in the like 80 million or something. It's a huge number. And then what Morgan just showed us, right, was that there's actually these crossing over events. And I think it's like 2 to 3 crossing over events on average between on each chromosome. And so, again, huge numbers, huge amounts of variation here. Okay, you get the point, right? Okay, sometimes things don't work right. Okay, so in meiosis, what happens if meiosis goes wrong? We get a situation called non-disjunction. So if homologous chromosomes don't separate normally during meiosis 1, or sister chromatids don't separate normally the way they should during meiosis 2, you can end up with this situation where you get the wrong number of chromosomes in those gametes, right, in the daughter cells. And so here we see a situation, right, where since these didn't split, these cells each ended up with 2 copies of that gene, and these cells ended up with no copy of that taller gene, right? And so either way, none of those gametes have the right chromosome number. So we refer to that as aneuploidy when you have the wrong number of chromosomes. Aneuploidy A. Right, or a cell is aneuploid. That's an O. Aneuploidy. Okay. That means the wrong number of chromosomes, right? And then the same thing can happen if that takes place in meiosis 2. We did end up with 2 cells that are normal, but this one has an extra copy of the chromosome, and that's lacking a copy of the chromosome. So the classic example of this is Down syndrome, right? And so this is also referred to as trisomy 21. And so here we see on the 21st chromosome, right, we've inherited a 3rd copy. And so that means that one of the parents donated an extra set of that chromosome, right? There was a non-disjunction. So this child either ended up, right, with one of the gametes like this or one of the gametes like this. And so there's an extra set of the chromosomes. And right, and so this is an example where there tend to be developmental disabilities. There's some very classic, physical features for this individual. But the real big problem here is that developmental disability. Now, often this happens due to non-disjunction, but I did want to, and this tends to increase with increased maternal age. So there's something going on where there's actually more non-disjunction events as the mother gets older. And we can talk about this more. It kind of makes sense because then the eggs are older, and it turns out there's actually a protein. When the chromosomes are being held together so that crossing over can occur, there's a protein holding them together. And what we'll see is that in older mothers, there's less of this protein, and so the chromosomes don't split properly. But This can also happen other ways other than non-disjunction. So there's all sorts of abnormalities, alterations that can take place in chromosome structure. So there's actually right? A chunk of a chromosome can get deleted or duplicated or flipped, which doesn't sound like it would be too big of a deal. This is what's called a translocation. And so what's happening here is we're actually moving a segment from one chromosome, right, and swapping it. I guess they're showing this one going over here. And so there actually is a translocation that can cause Down syndrome, where just an arm of that 21st chromosome ends up in the wrong place. And so it's called a Robertsonian translocation. And so you wouldn't get this classic karyotype where you can see the 23rd copy of the 21st chromosome, but the child would still have Down syndrome and all the symptoms that come along. Other aneuploidies. So these are two aneuploidies that occur on the sex chromosomes. And so Klinefelter syndrome is when a child inherits two copies of the X chromosome and a Y. So now you're XXY. And you're like, well, XX makes female and XY makes male. And basically what you end up with here, if you have the Y chromosome, you are male, right? You have that those genes that tell the testes to start developing, but basically they end up being a more feminized male. So you're going to see some patterns of female development along with male development. Then Turner syndrome. So this is an individual who only inherits one copy of the X chromosome. Which doesn't seem like it should be a big deal, right? Because females are already kind of tucking away half of their X chromosomes into bar bodies. But this is another aneuploidy that's actually going to cause severe physical and mental disability. So the other name for this would be monosomy X. You only have one copy, mono, of the X chromosome. There's a trisomy X, which doesn't seem to cause problems. So since a lot of you are interested in going into a medical field, I thought I'd point this one out as well. So here's another translocation, right, where we're swapping a portion of chromosome 9 and a portion of chromosome 21. And this, it's called the Philadelphia chromosome. That translocated chromosome is very much associated with certain forms of leukemia, right? So these problems at the chromosomal level can be quite problematic. Imprinting says it makes a difference, and this really only happens in a few traits in mammals, but the phenotype, right, that physical appearance, the outcome of inheritance, depends on which parent passed along the allele. And this is super strange. So in this example, basically what we see here, right, and this is a little confusing to follow, but if you inherit the normal gene, so this dark purple is the normal gene, if you inherit it from dad, you become a normal sized mouse. If the male, again, from the father, if you inherit the mutant gene, you become this dwarf mouse. So basically, for whatever gene this is, they're showing that mom's gene is the one not being expressed, right? And so whatever you get from dad is what's being expressed here. So again, genomic imprinting basically means that we're silencing certain genes depending on which parent passes them on. And this is really strange. Scientists are starting to think this has to do with something called methylation, so the addition of a methyl group, a CH3, that somehow basically like blocks the DNA from being used. So that would silence, in this case, mom's version. Anyway, there's a ton of fascinating stuff. Like if you're interested in this at all, you should totally check out taking a genetics class and really getting to delve into some of this, because there's so much that really in this intro course we just get to kind of skim over the top. It's kind of a tease. But there we have it. That is chapter 15. All right, I'll see you soon.